Hello, everyone, and thank you for attending this activity. My name is Maria Arcila, and I'm one of the founders of the Ibero-American Society for Molecular Pathology. This will be the first of several webinars that our society would be offering, and this is being presented across 3 separate virtual rooms in English, Spanish, and Portuguese. Recordings will also be available at our website to access for reviewing, which you can access through the QR code that you will see on the title slide and in the final slide on your screen in one moment. So our topic for today is the molecular assessment of early and residual disease in solid tumors using circulating tumor DNA. The access QR code for our website is at the top of the screen. In today's talk, we're going to discuss and refresh our knowledge on some definitions, review the current strategies being utilized for assessment, and also look at some of the challenges and opportunities in clinical applications. These are my disclosures. So let's begin. In the long fight against cancer, every patient wants to be declared cancer-free. But what does cancer-free mean? In the course of treatment of cancer and during cancer screening, we often hear the terms of a complete remission, no evidence of disease, no detectable disease. And why do some individuals with cancer and after treatment, they relapse just after a few months or years later, while others remain in permanent remission? And why, despite screening for cancer, patients often present at late stages? What is really a cancer-free state? And emerging evidence and research shows that the answers to these questions may be closely connected to the concept of molecular residual disease or molecular detectable disease. In the past few decades, we have witnessed the emergence of a new type of medicine, the personalized medicine, which hinges on the molecular underpinnings of disease. In cancer patients, molecular characterization is, of course, essential for multiple aspects of patient diagnosis and management, serving as the basis for tumor profiling and classification, risk stratification and prognostic assessment, as well as prediction of therapeutic response, residual disease assessment, and therapeutic monitoring. This is also presenting new opportunities for early disease detection and cancer screening. Let us focus first on residual disease. And let's just go over a few definitions. So for many years, we have used several approaches to determine the presence of both residual or recurrent disease, which includes imaging, histology, immunohistochemistry, and serology. But with the advent of molecular biomarkers, the concept of molecular residual disease has emerged as a potential tool that could be more sensitive. Importantly, in both solid tumors and hematologic malignancies, we often hear the acronym MRD, and the M in that MRD has been used interchangeably to mean either minimal residual disease or molecular residual disease, but these are not the same. And the actual definition depends on the level of disease and the disease context. So for the purpose of this talk, we're going to be talking about the presence of a very, very small number of cancer-derived molecules that remain in the patient during or after cancer treatment. This is a disease level that is not detected by imaging technology, and the patient has no clinical signs of lingering disease by any other ancillary method. Similarly, these very, very low detectable levels could be used for de novo early diagnosis, so the techniques that I'm going to be discussing from now on will pertain to both. Circulating tumor DNA holds substantial promise as a marker for residual disease and for early detection. As an early detection method, it would be particularly important for those cancers that do not currently have accepted screening methodologies such as ovarian, pancreatic, and gastric cancers. And you can use many features that are intrinsic to cell-free DNA that could be leveraged to detect disease, including mutations, copy number alterations, rearrangements, and fragment characteristics. Cell-free DNA testing may also be used synergistically with other multi-omic biomarkers to enhance the detection of disease. For instance, assays may incorporate proteins, epigenetic markers, circulating tumor RNA, nucleosomes, exosomes, and associated immune markers. Ample research is happening in this space, and many companies are currently competing to develop marketable solutions. Now, testing may be accomplished through the same technologies developed and utilized for tumor tissue diagnostics, with enhancements that try to push the limit of the technology to reach higher sensitivity and specificity. There are non-NGS approaches that you can see on the left of the slide that are extremely targeted, including real-time PCR, digital PCR, allele-specific PCR, and beaming technology. The Roche Cobas system, for instance, utilizes qPCR and is the first FDA-approved method for MRD testing in lung cancers. But the development now is more geared to the NGS technology using amplicon capture and hybrid capture technologies. Now, comprehensive approaches using whole genome and whole exome sequencing are now trending to increase sensitivity and specificity by allowing the assessment of other markers like methylation and fragmentomic profiles. I will obviously not be able to talk about all of these, but I will highlight salient technologies that are commercially available. An important detail as far as approaches go is whether the assay is tumor-informed or tumor-agnostic. Tumor-informed is based on prior knowledge of the tumor mutational profile at diagnosis, and typically tumor tissue is sequenced by whole generation sequencing or whole exome sequencing or a large NGS panel, and those variants are then monitored in the plasma by tumor-informed fixed or bespoke panels. The number of the follow-up variants can vary from 1 to 100, mostly based on investigator personal criterion or disease-pertinent bioinformatic algorithms. While these approaches is streamlined and cost-effective, a major pitfall is that you would be missing emerging or potentially informative targets, particularly those that are associated with clonal evolution and resistance mechanisms. Now, on the other hand, we have the tumor agnostic tests, and these are conducted without prior knowledge of the tumor profile. This may deliver lower sensitivity and specificity if they are designed only to detect mutations. There are also limitations in the specificity of these assays associated with overlapping mutations across tumors or the presence of coexisting clonal conditions such as clonal hematopoiesis. Newer assays now incorporate methylation status and fragmentomics to improve both the sensitivity and the specificity, with more details that will come later in the talk. Extensive research related to cell-free DNA and innumerable assays do exist. However, in this very busy table, I am summarizing the current NGS-based commercial platforms for tumor-informed analysis. These query single nucleotide variants, small insertions and deletions, and copy number alterations. Some assays may also detect viral targets, such as a NavDx assay, which detects HPV targets in HPV-driven tumors. It is important to mention that there is a wide range of claimed sensitivities, but ultimately the sensitivity of the assay is contingent upon the amount of cell-free DNA that is recovered from the specific sample of the patient, which could be highly variable. You may note that some of these assays are already covered by insurance companies here in the US. On the tumor agnostic side, there are some commercial platforms available, with Guardant Reveal as the only one that is covered by insurance in the context of colorectal carcinoma. So let's review some of the details and highlights of these assays. We have, for example, the Signatera assay, which is covered by Medicare for select indications, specifically for monitoring disease progression, disease recurrence, or relapse in patients with colorectal, bladder, breast, and ovarian cancers. This is also suitable for monitoring of response to immune checkpoint inhibitor therapy for patients with any solid tumor. This is a tumor-informed assay for point mutations and insertions and deletions with a maximum limit of detection of 0.001%. Multiple studies have used this assay to show that positive results of cell-free DNA, of circulating tumor DNA, can predict relapse with an overall positive predictive value of more than 98%. Studies also show that positive circulating tumor DNA results can be seen in patients with a lead time of 3 to 10 months ahead of imaging studies. Now, as an example, this is a study on bladder cancer. And as you know, bladder cancer is a common malignancy with more than 570,000 new cases each year worldwide. And approximately 25% of those patients that are diagnosed will have muscle invasive bladder cancer. And despite curative intent treatment with neoadjuvant chemotherapy followed by radical cystectomy, approximately 50% of those patients with localized disease will develop metastatic disease, and only 40 to 45% have pathologic response to the neoadjuvant chemotherapy. So in this context, early detection of metastatic disease and effective monitoring of treatment response are critical to improve patient outcomes. This study outlines the prognostic value of neoadjuvant chemotherapy treated patients with more than 5 years of follow-up after radical cystectomy. So this first figure represents the cumulative ctDNA status in patients at the time of diagnosis. What you can see in this Kaplan-Meier survival analysis is that both recurrence-free survival and overall survival for patients with positive ctDNA, which is depicted in red, have a significantly lower survival compared to those patients that are negative. Now, similarly high correlation is also seen after neoadjuvant chemotherapy, before and after resection, which you can see in the plots presented. And this is at the year mark after resection. Now, notably, even after 1 year after resection, about 20% of patients who recurred had no detectable ctDNA. And also 4% of patients with positive ctDNA status did not recur. So there is something more that needs to be done in terms of this sensitivity, and some of these may not necessarily be specific, or they actually need more time to follow up. But regardless, what has been shown is that ctDNA positivity has a median lead time of several months, and in this case, 4 months over radiographic imaging. And patients will have a sequential increase in ctDNA levels before their clinical relapse, and the speed of the rise correlates with the lead time in the patients that are being monitored with several time points of cell-free DNA sampling. Now, INVISE is another assay. This is a multi-cancer tumor-informed assay that is NGS-based, and it uses unique molecular identifiers, or UMIs, and updated anchored multiplex PCR chemistry from Archer Technologies. As an example, we have this study, which is a multi-center prospective study of patients with primary non-small cell lung carcinoma. And in the TRACER study, they enrolled 197 patients and tracked 200 tumor-specific mutations in more than 1,000 plasma samples. The aim of this study was to define the trajectories of lung cancer through multi-regional and longitudinal tumor sampling to determine if cell-free or circulating tumor DNA could serve as a phylogenetic biomarker in early stage non-small cell carcinoma. Now, the initial tumor in this study was characterized performing whole exome sequencing, and both clonal and subclonal mutations were tracked to infer clonal evolution. The Kaplan-Meier curves that you see here correspond to the preoperative ctDNA status. What you can see is that patients with ctDNA positivity were associated with a lower survival, but this was only clinically significant for patients with lung adenocarcinoma. The ctDNA detection was associated with clinically occult mediastinal lymph node disease, and about 97% of patients with positive results relapse within 1 year clinically. This is a super busy slide, but I do want to highlight some main points, and I will refer you for further details to the actual publication. Now, the plots here summarize the results of this cell-free DNA assessment and clinical evaluation across the evaluated points and patients. And the solid black dots represent ctDNA positivity. And the patients in these plots are stratified into 3 separate groups depending on their positive or negative results at 120 days after the surgery, which is the landmark point. And they are stratified into positive, negative, or unevaluable. Among those patients that are landmark positive at the top, which is 25% of all the patients, 93% of these patients relapsed at a median of 228 days after ctDNA detection. Among the negative landmark patients, 20% relapsed, but 84% of the patients that were negative ended up turning positive in the monitoring phase of the study by serial sampling. It is important to reiterate here that this assay claims a limit of detection of 0.008%. But to reach the sensitivity at this level, you need more than 30 nanograms of DNA input, which is often not possible in lung adenocarcinoma patients or lung carcinoma patients due to low shedding. So this means that not only numerous patients may end up having invaluable results because there is just no DNA recovered, or there is a low amount of DNA that cannot give you the sensitivity that you really want to acquire based on the assay that you're using. Now, next we have the Foresight Clarity assay, which is based on phased variant enrichment. The phase-seq is an analysis approach that increases background error rates, or actually decreases background error rates and increases sensitivity by requiring the concordant detection of 2 or more distinct somatic mutations within a single DNA molecule rather than just one unique variant. Most studies that have used phase-seq have focused on hematologic malignancies, but in this ASCO study that I want to present, which was presented last year, the approach was tested on non-small cell lung carcinoma. Now, the tumor-informed analysis was performed to compare the performance of 2 different assays. One, the single nucleotide variant assay, CAP-seq, and the phased variant assay, Foresight, on 269 samples from 46 patients. And they looked at the assay's ability to predict recurrence either post-therapy or post-surgical resection. What you see here in the Kaplan-Meier curves is that the disease-free survival. So with both assays, you see that the relationship of ctDNA positivity in red with inferior survival is similar. However, the phase-seq approach allows a much higher detection of low levels of circulating tumor DNA, which actually results in a more definitive prediction and further widening of those curves, as you see on the right. So a superior assay Notably, even with the most sensitive assay, there are going to be a few patients that recur despite negative results. And in this case, for the phase-seq approach, there were quite a few patients that were negative, and they still recurred. But what they noted was that these were patients that were enriched for CNS recurrences, which you would not be able to detect as easily on a plasma sample. And some of them were actually solitary lung recurrences that looked more like a second primer rather than the same tumor that the patients had initially. So in this study, the time frame to recurrence from landmark was distributed broadly between 0 and 24 months. And you can see the summary of the patients that ended up having negative cell-free DNA result or circulating tumor DNA results, but who recurred. And you can see that they're enriched again in CNS recurrences and lung recurrences with different profiles, indicating that they were perhaps a second tumor. They did do additional comparison with other approaches that use single variant, including Signatera, Radar, and CAPS-seq, which I just showed. And this supports that the phase-seq approach has a much higher detection of residual disease compared to the other assays. Moving on to Garden Infinity, this is also a tumor-informed assay that uses both mutation and methylation and is currently only validated for colorectal cancer. But there are clinical trials that are ongoing for lung cancer, breast, and pancreatic cancer. Now, methylation assays require a lot of DNA, which is not possible in plasma samples. But this assay uses a methylation enrichment technology that offers up to over 1,000% increase in molecule recovery compared to the bisulfite method, which allows a much more robust assessment at the end with a much smaller samples. In this study, patients with colorectal cancer who had circulating tumor DNA tested at 1 month post-therapy and followed for more than a year demonstrated that the utility of the circulating tumor DNA testing with this assay, showing similar results to prior assays in that a positive ctDNA result after completion of the therapy actually recur. Now, one of the things that is important to mention here is that the use of both mutation and methylation targets deliver much higher sensitivity compared to either mutation or methylation alone. So you can see here on the light blue, this is mutations only. On darker blue, the using methylation, and then using both at landmark, longitudinal, and on the overall surveillance of the patient, using the two approaches together is going to be much better in terms of sensitivity. Now, there are numerous other studies that document the same experience for disease monitoring. And I referred our attendees to this publication specifically, that is a very, very good summary of current studies. But with all of this information that I have provided and what is already in the literature, the question, an important question is, are we ready for a new classification beyond the TNM classification that now incorporates blood as part of the assessment? So the reality from these studies and all of the compiled data is very, very promising. Both detection of ctDNA at diagnosis and after treatment is associated with disease recurrence and decreased overall survival. The sensitivity varies according to the assay and the disease type, from 50 to 60%, but the results have a very high positive predictive value that is very close to 100%, and the median lead times are generally several months ahead of imaging. Studies that provide evidence to support routine ctDNA remain small and limited in scope. So there we require validation using much larger independent cohorts to be able to make this a routine assessment. Now, there are many challenges to being able to provide this routinely to patients from biologic, logistical, technical, and regulatory in nature. So from the biologic point of view, there are numerous variables affecting shedding and the amount of ctDNA in circulation that could lead to false negatives. Also, plasma reflects all parts of the body, so there are confining factors related to co-occurring clonal and neoplastic conditions, which are a source of false positives. There can also be major differences in local recurrence versus distal mets, including the emergence of new clones, which when they're just emerging may not be equally captured in these assays. Now, logistically, the coordination of timing of the sample, enforcing proper collection and delivery, testing implementation, this is all operationally very intensive. There also has to be very high technical expertise, which may not be available in all settings. There are numerous pre-analytic, analytic variables and technical considerations that one has to pay attention to, including the timing of the collection, how much volume can be used and collected, the storage, the processing, the testing resources, the sources of artifact, the analysis, the number of genetic targets that you would be using, the methods for correction, the bioinformatics support, and also the support that you would have for artificial intelligence and machine learning. Finally, of course, there are the regulatory and finance parts of this. At this moment, there is no standardized method to use, and you can see there are multiple different assays that could be used that use different technology. And there is lack of validated large-scale clinical trials that say that this is something that needs to be implemented. Many assays, of course, with all of this, are not covered by insurance companies. But there are numerous opportunities in the horizon, not only for the detection of cell-free DNA in plasma, but also cell-free DNA from many other fluids, all of which are suitable for assessment and some highly enriched in tumors. So, for example, cell-free DNA in tumors affecting the central nervous system can have much more tumor than a biopsy itself, and could be even better than for any type of testing compared to the tumor tissue. Also, saliva for head and neck cancers could be very valuable as well. There are also numerous opportunities in other biomarkers beyond mutation and methylation that would expand the utility of the assays, not only for higher sensitivity, but for higher specificity, which is important in cancers at early stage if you don't have a biopsy that can inform the mutational profile. So we talked about whole exome, whole genome, and methylation, but I want to highlight a little bit the fragmentomics, which is gaining significant momentum. Fragmentomics is the study of fragmentation patterns of cell-free DNA, and it analyzes many aspects of cell-free DNA beyond the size of the fragments to understand other characteristics such as topology, nucleosome footprints, preferred ends of cleavage, and genomic coordinates, nucleosome, the end motifs. So there is a lot of information that you can gather from this. Importantly, for instance, obviously this fragmentation is not a random process. There are numerous factors that affect those little fragments, including the chromatin and the nucleosome structure, the nuclease activity, the DNA sequence, and the available cleavage sites, as well as the DNA methylation status itself of the fragment. but establishing a pattern that can help identify the tissue, that it could help identify the tissue of origin when you're testing cell-free DNA from plasma. So, for example, the nucleosome footprint analysis is an emerging and incredibly important measure to infer gene expression. Very briefly, depending on the tissue of origin and the tumor type, there will be specific regions of the genome that are actively being expressed. So when you do a low-pass whole genome sequencing of cell-free DNA, you can see that there is a distinct drop in coverage in areas just upstream of the transcription start sites of highly expressed genes, which is correlating with the areas of naked DNA that are not protected at that point by a nucleosome. This gives rise to a periodic oscillation that you can visualize when you do the pileup of sequencing data. So what you can see is, again, these oscillations of high coverage versus low coverage and high coverage versus low coverage. And this basically is a surrogate for expression of specific genomic regions. Now, by contrast, on the right side, areas that are quiet in the genome will have no oscillations and have all of the fragments would have different coordinates. Now, similarly, one can infer the methylation status of the cell-free DNA fragments, and that is because the methylation status of the DNA at CpG sites is quite particular. It turns out that fragmentation around CpG sites is about twofold higher if you have a methylated cytosine within an 11 nucleotide distance. And this is used to measure the cutting preference of the DNA at a molecular level. If the cleavage profile favors a specific cytosine, you can analyze motif ratios and infer the methylation or no methylation at that site. What is quite interesting is I want to show you the comparison of patterns that are derived from cell-free DNA from fragment analysis versus the standard bisulfite method. And what you can see that this correlation is quite amazing. You can, of course, see how this would have innumerable applications for tissue of origin classification. Now, on this slide, you see the bisulfite sequencing on top and the whole genome sequencing with the low-pass whole genome sequencing with analysis of the fragments on the bottom. And again, you can see the very, very high correlation. Now, to robustly scrutinize the characteristics of cell-free DNA molecules and the vast amount of information that is derived from high-throughput sequencing, genomic, epigenomic, transcriptomic, and fragmentomic data, one needs to use artificial intelligence and machine learning to enable the analysis and integration of these complex and high-dimensional features. And there are now several machine learning algorithms that exploit this cell-free DNA fragmentation features, including the Delphi algorithm and Fragma, for example. So I want to show you how one company is actually making use of this, and this is Delphi, which stands for DNA Evaluation of Fragments of Early Interception. And it's a product that was developed by Johns Hopkins University and licensed by Delphi Diagnostics for commercial use. Now, Delphi Diagnostics is marketing the First Look lung test. And this is an accessible way to enhance screening rates for lung cancer, which often fall behind other cancers for screening. And doing a simple blood test could encourage more patients to not only get screened, but to receive follow-up low-dose CT scans when necessary. Recently, the data for the clinical validation of the assay was published, and I will refer you to a lot of the details in terms of the eligibility criteria and the assignment of evaluable participants to the classifier for this assay, but they included in the classifier training 576 patients. So briefly, what they demonstrate is that genome-wide fragmentation profiles are altered in patients with cancer, and this reflects the underlying chromatin structure. In this figure, what you can see at the top is that is the 395 patients that do not have cancer. And by contrast, the patients on the bottom, 181, are patients with lung carcinoma. So you can see the difference. There is much more fragmentation, which could be used to infer whether it's disease or not. They further demonstrate that in patients with known cancer, detection was much higher in patients with small cell lung carcinoma compared to non-small cell lung cancer. And that detection is also higher in patients with higher TNM status, higher T, higher N, and the presence of metastasis. Of course, this translates into a sensitivity that is much higher for patients with stage 4 of about 98% compared to 71% for stage 1. But even with a sensitivity of 71%, this is much higher than other methods of assessment. The authors in their publication make the point that at present, lung cancer screening rates in the general population are very, very low. And it's about 6 to 10% for eligible individuals. The incorporation of a blood test has major implications, and the authors propose an algorithm of cell-free DNA first, followed by low-dose computed tomography. In this approach, they consider two different scenarios of simply screening 10% of the eligible population versus 20%, which they denote here as low uptake or high uptake. So let me just walk you through this slide. The gray is the computed tomography. The low uptake is the blue, the light blue, and the high uptake is the dark blue. And so what they show is that at a 5-year period, the results would not only show a much higher prediction of cancers on the number of cancers that are compared to low dose CT, but also the number of predicted cancers at stage 4 will decrease while increasing the detection of stage 1. So this right here is the stage 1. So you increase the stage 1, decrease the stage 4, which is what you actually want to do, detect the disease a lot earlier. This also predicts a decrease in lung cancer deaths. And importantly, just that one simple blood test could significantly decrease the number of CT scans that would need to be performed to detect one single lung cancer. So it comes with additional savings. So there is a lot more to talk about, but we are at the end of our presentation. So there are some take-home points here, which are that in the fight against cancer, accurately assessing residual disease status and early detection are critical. That liquid biopsies have emerged as a valuable tool for detection of cancer that is not invasive, is also easier and more democratic for patients who do not have access to care. And also, detection of ctDNA after treatment and with curative intent is associated with disease recurrence and decreased overall survival. Now, although patients with low-level disease usually have lower levels of ctDNA, the studies do demonstrate that positive results often precede clinical manifestation by weeks to months and are highly, highly specific. As other game-changing diagnostic technologies, there are many, many challenges to overcome, along with numerous other opportunities for future development and discovery that we will need to explore. I thank you very much for attending this lecture. We'll leave you with our QR code again. This will take you to our website, Asociación Iberoamericana de Patología Molecular, AIPM. Please check us out and become part of our society. Also, you can receive updates on future conferences and webinars and educational opportunities that we're very happy to offer to our community. Thank you so much. Bueno, entonces no sé si estoy al aire para contestar preguntas. So we have a few questions. I just need to make sure that people can hear me. So I need somebody from online. Oh, hey, Mariana. I don't think I can hear you. Your microphone is off. You're still off. All right, so I think we have a few questions from the audience. So I'm just going to, because we have different people from who are Spanish speaking and English speaking. I'm just going to do both in English, but I'm happy to respond to these questions online as well or through email. But so one of the questions that I had here from Lucia Kisan is what is the influence of chemotherapy and radiotherapy and how much ctDNA you can measure? So I think I mean, this is a complex question because it really depends on the intensity of the chemotherapy or the radiotherapy. And one thing that one has to understand is that at the time that you're giving the treatment, you're not just treating the tumor, but you're also treating the normal cells around it. So you can actually have a much higher amount of cell-free DNA that can be recovered from the sample when you have immediately after treatment, which will take some time to clear off. But not necessarily all tumor DNA. So it could be that there is a lot of tumor and different proportions of tumor DNA in the entire context of all of the cell-free DNA that is being released. But the answer is you will have much more of both. There are, depending on the intensity and the degree of damage that you're causing, there is going to be a lot of cell-free DNA, much less. And the same way that there is a certain amount of cell-free DNA that increases after you do a surgery, for instance. So there is a period of time that you actually have to wait before you can actually measure and recognize that one is damaged, and then after a certain period of time is going to be cell-free DNA that is residual. So the timing of a liquid biopsy in different situations is something that has not been yet worked up. I hope I answered that question. So the next question comes from Eddie Veronica Mora Gonzalez, and who wants to know about single nucleotide variants and which one of the platforms that I just mentioned are better. So there are 3 or 4 platforms that only use single nucleotide variants, and in general, the sensitivity of these platforms is between 0.01 and 0.001%. But again, one of the key features is not necessarily the platform itself, is really how much cell-free DNA you're recovering from the patient. So if you think about it, if I have a patient that has very, very minimal residual disease, the amount of cell-free DNA and circulating tumor DNA may be very low, so that in the laboratory, what you can recover from that sample could be just a tiny little bit of DNA. And even if the test itself can get to a sensitivity of 0.001 by I only have 2 molecules, there is no way that I'm going to get to that sensitivity because you need the amount of template. But any of the platforms that I talked about that are single nucleotide variant, either Radar or Signatera and CapSeq probably is the one that gets to the highest sensitivity. There was an additional question on what is the maximum amount of time that you can save a plasma sample to make sure that it actually works. And this is when you refrigerate it at -20. So I can tell you that in my own experience in our laboratory, -20 doesn't necessarily protect the DNA as much as -80. So we try to keep all of our samples at -80 to be able to maintain the cell-free DNA intact. But we have had samples for a year, for 5 years, and we're still being able to use the samples. But again, we keep them at -80 rather than -20. Um, the last question that I have here from Carlos Mauricio Figueredo is, I want to know which ones of the platforms can you actually do fragmentation? So to be able to analyze the fragments, you need to do whole genome sequencing. And you have to do low pass whole genome sequencing to be able to get all of the genome and get all of the information that you need. The very, very targeted panels are not good enough. It has to be a low pass whole genome sequencing, and you could use any type of whole genome sequencing as long as you're doing that comprehensive assessment. And then the fragmentation analysis gets done by bioinformatics and machine learning and artificial intelligence algorithms that I talked about. I am looking for more questions in here, but I'm not seeing any more, and we are actually at time. But I will be happy if anybody has additional questions. You can either forward them to me on an email, or I'll be happy to answer any other questions or get in contact with someone from our team. All right, I'm checking a few more questions that are arriving here. I actually on my iPhone, so give me one second. So there is one question about in prostate cancer, can you do okay? So in prostate cancer, can you do these? Yeah, can you follow up this patient, these patients with prostate cancer? And it really depends. Prostate cancer is actually incredibly difficult to follow because a lot of the changes that happen in prostate cancer, they are better done with methylation analysis. But as long as they have certain mutations, you can actually do the follow-up. I'm not exactly sure that I understand the entire extent of the question. But and I don't see Mariana online that can actually maybe answer this question as well. But I think that as far as questions go, I'm not getting anything else. So and I'm not, Mariana was trying to, it was going to be the moderator, but she had a technical issue with her connection. So I think that we are going to, if I don't get any more questions online, I think we're done with our presentation. I do want to thank you all for attending. It's late in here, and I know that it's equally late for a lot of you people that are joining us from other countries. So thank you so much. We hope that this is going to be one of many more webinars that we're going to offer, and we're taking ideas on what you actually want to see and how can we as a society serve you better. So become active in our society, because I think it is important for all of us to be involved to make sure that we move molecular pathology forward in Latin America. So thank you so much all for attending, and we will see you on the next webinar. Thanks again.